biotechnology involves an important technique which forms a part of the genetic engineering. Now we have understood, or I have talked about in the first video of biotechnology, an introduction, that biotechnology involves the formation or achieving the rDNA or the recombinant DNA, followed by the cloning of that particular gene and that transfer of that particular gene into a suitable host for the cloning, that is gene cloning and then the gene transfer. These are important steps of the biotechnology. Now when we talk of recombinant DNA is the molecule of DNA obtained from two different species. These are desirable DNA areas or regions which are obtained from the two species, which are then inserted into an host organism to produce genetic combinations, and that are then used. These genes are then allowed to produce the proteins which are then used in science, medicine and agriculture and various fields. So the genetic engineering forms an important part of the formation of the recombinant DNA. Now, when we talk of recombinant DNA, the first rDNA was produced by Stanley Cohen and Herbert Boyer. So the first rDNA was produced by Boyer, that is Herbert Boyer and Stanley Cohen. These two scientists first produced the recombinant DNA at Stanford University, and they used the plasmid of Salmonella typhimurium. Okay, now let us talk about the genetic engineering, what it is actually. Now genetic engineering enables precise manipulation of the genetic material so that desirable changes are observed. So here when we are talking of the DNA, that is the recombinant DNA, the recombinant DNA has to be a DNA whose desirable traits we want to take. So therefore it involves the precise manipulation. Now the problem is, if we simply introduce a foreign gene into a host organism, then it will not be inherited by the progeny of the host organism. Like we have isolated the DNA which is carrying the desirable trait and introduced into the host organism. But since this DNA is not a part of the organism's genome, it will not be replicating or it will not be inherited by that host organism. So for it to be inherited, it has to be integrated with the genome of the host, because what we are doing is we take a plasmid which is an extra chromosomal genome, right? It is an extra chromosomal genome, but if we introduce a fragment of the DNA, since it is not forming within the cell, it is not forming a part of this genome, it will not be replicated. This is foreign DNA which we have incorporated. And this is the plasmid. So since it is not forming the part of this, it will not multiply. So what we need to do is we have to put it in a DNA sequence where the replication takes place, and that is the ORI site. So this foreign DNA has to be integrated at the ORI site or the origin of replication, and ORI is responsible for initiating of replication. So for the multiplication of the alien pieces, of DNA, it should form a part of the chromosome, and that necessarily should be the ORI site. Now, since the alien DNA is linked with the replication, it replicates and then it multiplies in the host organism, producing similar genome or clones. So once this foreign DNA is incorporated within the ORI site, then it will start replicating. And once it starts replicating, it will form the clones. And this process is known as gene cloning. That is, forming multiple identical copies of any template DNA, that is recombinant DNA. Now when we talk of the biotechnology, apart from various uses, there is an production of insulin, then the polio vaccine. These are some interesting examples. The polio vaccines. Polio vaccines are actually the weakened or killed pathogens, and these are then introduced into the body. But what can be the risk? Suppose if even one virus is not killed, that or it is not weakened enough, then it will be able to cause the disease, or it will affect the human body. Now, actually, what is responsible for causing the infection? It is the protein which is present on the capsid, that is the outer wall of the virus. So here, what are we introducing? We are introducing the protein present on the capsid is isolated, which acts as an antigen, and it is introduced into plant like banana, right? What we have done, we have taken the capsid of the virus. Capsid is that protein which is proteinaceous covering, and it is responsible for causing the infection. So this capsid is removed, capsid of virus, it is removed, it is introduced into plant, and when it is introduced into plant, translation and transcription will take place. That is, first the transcription and the translation. Antibodies will be produced, and the edible vaccine is ready. There will be no chances of person who is taking this banana will be getting the infection by polio virus, because here the chances will be nil over here, without any risk. Another interesting example is that of the firefly. Now, what have we done in the case of firefly? You know, the firefly glows at night, and it is due to a protein called as luciferin. Luciferin is a protein that is present in firefly, and this is responsible for the luminescence during the night. Now, this protein is extracted, the gene which is responsible for this protein is extracted, isolated, and introduced into plant. Now, the plant will gradually incorporate this gene, and this gene will multiply, and the plant will develop the ability to absorb the luciferin. Since it is present over there, this will absorb in the plant also. This will absorb the light during the day, and during the night it will emit the light. So we have plants that will emit light during the night, and these plants are planted again along the roadside. So the plants glow at night. These are some interesting examples where we have put biotechnology into use. Biotechnology can be applied in other areas such as healthcare areas, the crop production, agriculture, then other uses of crops, and for producing the biodegradable products such as plastics, biofuels, etc., and other plants, that is genetically modified crops, that is GMO crops and organisms. So we have various applications of this biotechnology.